Hi, we are going to dissect Cassiopea medusa, the upside down jellyfish. First we'll have a look at the general features that we can see externally, and then we go on through the dissection and see other internal features. Now we are going to proceed with the animal observation, and it's good to have a bright or a dark background because it will highlight different structures. So this is a white plastic tray, but we can use a plastic sheet to contrast some structures. Okay, this is good to visualize some features of the animals. The first thing to look at is to check the margin of the animals. This is a feature of the Scyphozoans. If they have a lobulated margin, this is characteristic of the group. And also we can check if they have sense organs or rhopalia on the margin. So we can see some rhopalia at the margin. In Cassiopea we might have 16 or more depending on the species, or sometimes 12 if it's Cassiopea frondosa. This is the upside down jellyfish, and as the name says, it usually stays with the exumbrella laying on the bottom. So we are seeing the oral surface, and then I will turn the animal, and this is the aboral surface or the exumbrella. We can see a faint line here that lines the stomach. We'll make a dissection and then we'll see. And in this, near the margin, we barely see a contour that it's called Hingwurst. We can see some furrows or grooves in this area. Okay, so this is the exumbrella, and this part is the subumbrella of the animal, the oral surface. In the oral surface or the subumbrella, we can clearly see the musculature here that are in feather-like pattern. We can see the bases of different oral arms, and we can see between two oral arms we can see a round hole that it's the subgenital pit. And the oral arms in the Rhizostomeae in general, they are 8 oral arms. We can see it has some branches, and at the tip of each branch there are small mouth openings. In many of the rhizostomes, but it's more easy to see in Cassiopea, we have a lot of appendages. In this genus they are harboring zooxanthella symbionts in this part. Here we have the marginal lobes, and the sense organs are here. It's not so easy to see it. Now I will open the animal using the pillars between the oral arms. One good thing to do is to cut near the subgenital ostium, so we remove completely the oral disc. By doing this, we will expose the internal structure. We have to remember that the mouths on the oral arms are all connected to small canals through the oral arms, and then they will reach this central stomach that I'm exposing right now. Now we have two parts, the umbrella and the oral disc, and we can see here now we can see better the arms. So we have 1, 2, 3, 4, 5, 6, 7, 8. 2 were removed. And this part are the gonads and the gastric filaments. They are folded many times here. As we can see, the stomach is a round structure. So the gonads are inside the gastrovascular cavity that we are calling stomach. If we look at the umbrella, the bell of the animal, we'll see the hole that forms the central stomach. Then I will show that the central stomach is connected to the margin through many canals of the gastrovascular cavity. In this part we have the gonads and the gastric filaments. If we are going to look at the gonadal tissue, we can just take a little piece of this, put in the microscope slide, and then check if it's a male or female. Now we can take a piece of the gonads with a forceps, and then we put it in the microscope slide, and then we finish the preparation in the microscope to observe if it's a male or a female. Now we are going to inject our color dye. You can use everything you have. I'm using methylene blue, but you can use even a cake coloring, anything that you have at hand, just to highlight the canal system. If you have a living animal, you can use meal. So I'm having a syringe with the methylene blue, and then I will try to insert the needle at the base of a canal. It's good to have the dark background off, so it will be easier to see the animals. Okay, then at the base or at the sides of the stomach, you can see the color in the canal system. With this we can see the canals that are connected to the central stomach, and they go through the margin, reaching the sense organs and so on. And this is important because different groups of rhizostomes have different shapes and organization of the gastric system.